Hello and welcome to the webinar. I'm Ann Thayer, contributing editor to the C&EN Media Group, and I'll be moderating today's event. This webinar is entitled Fast Multinuclear Benchtop NMR Spectroscopy, How the Highest Sensitivity Saves You Time and Enables New Applications, and it's being sponsored by Magritek. C&EN works with sponsors to identify topics of interest and value to C&EN's audience and consistent with C&EN's mission to provide news and analysis of the chemistry enterprise in a timely, accurate, and balanced fashion. During the webinar, you can adjust the size of the slides on your screen by grabbing the lower right corner with your mouse. If you need technical assistance, please look at the Help widget at the bottom of the screen or type your query into the Q&A box. If you're disconnected during the webcast, please log in again according to the instructions you received earlier. You're encouraged to contribute to the success of this webinar by asking questions at any time during the presentation through the Q&A box on your screen. The questions will be answered at the end of the presentation, and as your moderator, I'll be posing as many as time permits. Any questions that we don't get to, the speaker can follow up with you later. Please note that C&EN does not endorse any company, products, or services that may be mentioned in the webinars, and that each webinar will be archived at C&EN Online after the live webcast. The presentation today is being sponsored by Magritek. Founded in 2004, Magritek is a leader in manufacturing cryogen-free benchtop nuclear magnetic resonance spectrometers for the analytical instrument market. Magritek's revolutionary 90, 80, and 60 MHz Spinsolve family of benchtop NMR models offer the highest sensitivity and resolution available in the market. These portable systems are robust and easy to use, allowing modern NMR methods to be performed on the chemistry lab bench or inside the fume hood next to a reactor. Magritek has offices in Germany, the US, UK, and New Zealand, as well as a worldwide network of partners to help support its customers. During the presentation today, we'll hear from Andrew Coy, CEO of Magritek. Andrew has been with the company since its inception 17 years ago. He has a PhD in physics from Massey University in New Zealand, where he worked on pulsed field gradient NMR with Professor Sir Paul Callahan. I'll now hand things over to Andrew.
Well, thank you, Anne, for the kind introduction, and welcome everyone to this webinar about fast multinuclear benchtop NMR spectroscopy. I want to show you the latest developments and applications in benchtop NMR. In particular, I want to discuss how the sensitivity and performance of a benchtop spectrometer is critical to enabling new applications and features. Magritek has been developing cryogen-free permanent magnet NMR instruments for 17 years. In 2012, we introduced our Spinsolve range of benchtop NMR spectrometers, the first benchtop NMR to use standard 5mm NMR tubes. We now have around 1,000 installations of Spinsolves globally. All the spectrometers are manufactured in Germany in our 2,000 square metre purpose-built factory, shown here. If you browse the literature today, you'll find some amazing examples of Spinsolve spectrometers being used by chemists in some interesting setups. On the Magritek website, we have a list of over 200 journal references where a Spinsolve spectrometer is being used. A critical feature for many of these applications is that the Spinsolve can be placed directly in a chemistry lab, next to the reaction in the fume hood or near the sample where the chemist needs it. Being able to place the Spinsolve in the laboratory is possible because of the benchtop size cryogen-free permanent magnet used in the spectrometer. This enables a chemist to get their NMR spectra quickly and at a reduced cost compared to high-field NMR. The simple operation of Spinsolve with intuitive software means any chemist can operate the instrument easily and safely themselves. On the left is a list of some of the applications for benchtop NMR. In this webinar, I'm going to show some specific examples from the ones highlighted in green. In each of these application areas, you will find a wide range of possible samples with different molecules and different concentrations. In some applications, we are measuring NMR nuclei other than proton, such as carbon-13 or silicon-29, which are harder to measure. This brings us to a very important question that many chemists ask. Will benchtop NMR work for my specific application? The answer to this question depends critically on the performance of the spectrometer and the specific details of the sample being measured. A spectrometer with higher performance will be able to detect and resolve larger, more complex molecules with lower concentration because it has more sensitivity and more resolution. Both the sensitivity and the resolution depend directly on two fundamental properties of the magnet. First, how homogeneous or uniform the magnetic field is, often measured by the line width, and second, the magnetic field strength, which is often measured by the megahertz, the frequency you're working at. As you can see on the right, I have a series of NMR spectra acquired on the same magnet, but at the top we've deliberately made the homogeneity poor, and then as you go down to the bottom, the homogeneity gets better. With the poor homogeneity on the top, you can see the peak is smaller, and we can't resolve the two separate peaks. At the bottom, the homogeneity is much better, and the peak is both higher, and you can resolve the fine details and see the two separate peaks. So this shows that both the sensitivity and resolution are improved when we increase homogeneity. This discussion brings us to a very important difference between high-field superconducting magnets and benchtop permanent magnets. In high-field NMR, the magnets are large enough and the technology mature enough that all magnets, irrespective of their field strength, can be manufactured to be very homogeneous and uniform. This means it's almost always the field strength that defines the performance. A higher megahertz has higher sensitivity and resolution. However, in benchtop NMR, the situation is very different. Benchtop spectrometers have to be small and light enough to be able to fit on a bench or in a fume hood. However, as you shrink the magnet design, you also shrink the homogeneous region, and so the homogeneity over the sample volume can suffer. This means it is homogeneity that has the most significant effect on performance. A benchtop magnet design must first get the homogeneity under control before you can consider increasing the field strength. So in our benchtop NMR design, we have a delicate trade-off to make. We want the strongest magnetic field, but with the highest possible homogeneity over our 5mm NMR tube sample. At the same time remaining a benchtop size and weight, and it needs to be stable to changes in the environment. For Spinsolve benchtop NMR spectrometers, we use a patented high-efficiency modified Halbach that allows us to maintain a benchtop size and weight while achieving an excellent homogeneity and stability. The standard way to measure the homogeneity is to take a single scan of dilute chloroform and measure the line width in the middle and, most importantly, at the base of the line. In this spectrum of dilute chloroform, you can see the excellent homogeneity achieved, where we measure not just the line shape at 50% and 0.55%, but also at 0.11%, as is typically done on high-field NMR spectrometers. One of our golden rules we follow in magnet design is we do not increase the magnetic field unless we can maintain the homogeneity specifications of the magnet. Otherwise, you have the contradiction of a higher field NMR instrument with lower performance than an instrument with lower magnetic field. Another significant performance advantage of excellent homogeneity is the ability to do effective solvent suppression. As benchtop NMR spectrometers are deployed in chemistry labs, many samples and most reactions will already be using a protonated solvent. To measure these samples, we need to be able to suppress the very strong NMR signals that arise from protonated solvents. However, solvent suppression only works well if you have a very homogeneous magnet. There is an ASTM method to specify solvent suppression performance, which measures the anomeric protons of sucrose dissolved in water. In this spectrum, we have a sample of 20 millimolar sucrose dissolved in normal protonated water acquired on a Spinsolve 80 Ultra, both with and without solvent suppression. Without solvent suppression, the anomeric peaks are affected by the large tails from the water peak. However, with solvent suppression, the water peak has been reduced so much that the sucrose peaks are completely separated from the water peak. To really appreciate the performance of solvent suppression on the Spinsolve 80 Ultra, I have here a sample of 10 millimolar histidine in normal protonated water. In this first spectrum, the data is shown full scale, so you can see the full water peak before and after solvent suppression. Notice that the histidine peaks are so small they are not visible at all on this scale. In fact, we need to zoom in 4 orders of magnitude, or a factor of 10,000, to see the 10 millimolar concentration peaks. Notice again, several of the histidine peaks are affected by the large tails of the water peak. And notice again how, with the solvent suppression, these peaks are all cleanly resolved and well separated from the suppressed water peak. That water peak is now taking up less than 3 Hz of our spectrum bandwidth. A nice example of how better solvent suppression performance enables new applications can be shown using glucose dissolved in water. Glucose has both an alpha and a beta anomer. With effective solvent suppression, the anomeric peaks of the alpha glucose anomer are well separated from the residual water peak, even though they are closer to the water than the peaks of sucrose we saw earlier. But with this excellent solvent suppression, we can also see the peaks of the beta glucose anomer, which are visible to the right of the water peak. We could use this additional information to quantify the ratios between these two conformational anomers. We can also now measure the splitting of the doublets of the two anomers. The alpha glucose peaks have a splitting of about 2.7 Hz, which is due to the 60-degree dihedral angle with the non-equivalent protons on the adjacent carbon. Contrast this with the beta anomer, where the doublet splitting is around 7.2 Hz, resulting from the dihedral angle of 180 degrees. Martin Karplus described the correlation between J-coupling and dihedral torsion angles in the 1960s and would go on to win a Nobel Prize in 2013 for this work. We saw before how excellent homogeneity is the foundation for both good resolution and sensitivity in a benchtop NMR spectrometer. Sensitivity is very important because the NMR signals we are trying to detect are very small and can easily be lost in the background noise. With higher sensitivity, we can measure quicker. A 2 times higher sensitivity allows us to measure 4 times faster. We can measure samples with lower concentrations or detect impurities present at a low level. We can measure nuclei that are themselves less sensitive and less abundant, such as carbon-13, which is about 10,000 times less sensitive than proton NMR. We can also use diffusion NMR methods, which work by making the signal smaller with increasing field gradients, and we can make use of advanced methods like NOAA and NUS that themselves enable us to speed up 2D acquisitions. The standard way to measure sensitivity on a benchtop NMR instrument is to take a single scan of 1% ethylbenzene and compare the signal of the highest peak in the quartet with the noise as I've shown on the spectrum on the right. In this timeline, we illustrate the continuous development of Spinsolve since we introduced it in 2012. You can see here how our magnets have both increased in field strength and in parallel become more and more homogeneous, with the development of our ultra range of high homogeneity magnets starting in 2017. In 2014, we introduced the first double resonance probe, which allowed us to offer the first carbon-13 capable Benchtop NMR, taking advantage of the sensitivity and performance of our magnets. Just a few months ago, we introduced our new Multi-X probe. This next generation double resonance probe includes a set of electronic switches that are used to retune the probe completely automatically so that multiple X nuclei can be selected on the second channel. I'm going to show later some examples of how this new technology, combined with the NMR performance, opens up new application and improves the throughput and speed of the Spinsolve instrument. We're also very excited just last month with the launch of our new Spinsolve 90 with a 90 MHz magnet. With the Spinsolve 90, we've maintained the field homogeneity while increasing the magnetic field to 90 MHz. This not only gives us better chemical shift resolution, but also improves the sensitivity specifications, shown here to be greater than 240 to 1 on that 1% ethylbenzene sample. This is now the highest available sensitivity on the market today. A simple way to illustrate the higher performance of the Spinsolve 90 is to compare spectra acquired on different Spinsolve models of the same sample, but plotted here in Hertz rather than normalized to a ppm scale, as most of us are used to. In the stack plot here, we've acquired a spectrum of 250 millimolar ibuprofen on Spinsolve models with 43 MHz, 62, 81 and 92 MHz frequencies. The stack plot shows how the increasing frequency increases the chemical shift dispersion, spreading the spectrum out over a larger frequency range. If I now plot the same data on a ppm scale, the scale that we're used to, it demonstrates how the multiplets get more compact as the frequency is increased. This is simply because the splitting of the multiplets is independent of the magnetic field. Let's take that same spectrum and zoom in and look first at this non-net multiplet here at 1.8 ppm. You can see as the magnetic field is increased, this multiplet here becomes more resolved. As well as the better resolution of the multiplets, these spectra have been normalized so the peak heights are all the same. And you can see now how the noise of the spectrum is getting smaller and smaller as the frequency is increased. So this demonstrates not only the increased sensitivity performance, but also shows that we're properly meeting our magnet design goal that I mentioned earlier. So now let's turn to a larger molecule with a more complex structure to demonstrate the benefit of the increased performance on the Spinsolve 90. The molecule here is brucine, which has a molecular weight of almost 400 Daltons, and the sample has a concentration of 250 millimolar, a typical concentration available to a chemist. The 1D proton is acquired in a single scan and shows a very high SNR. You can see almost no noise in the baseline here. We get very good resolution of some chemical groups, and there is some other groups that are overlapping. Now with this cozy spectrum on the right, we can resolve the couplings between these groups in a very useful way in a 2D spectrum that was acquired in just 13 minutes. Some of the couplings that are visible are highlighted here with coloured arrows. In the 1D carbon experiment here, we can clearly resolve the resonance of all the carbon atoms, although note that these two carbons have frequencies that sit on top of each other, so there's 22 carbon peaks in this spectrum. The concentration of 250 millimolar is quite low for carbon-13, but we were still able to acquire a very nice spectrum in 120 minutes. To really take advantage of the carbon resolution and using the proton sensitivity, the NMR methods of choice are the HSQC and the HMBC. The HSQ-CME spectrum, shown on the left here, was acquired in just 8.5 minutes. And you can clearly resolve all of the chemical groups except for the 7 quaternary carbons. And there's a very nice signal to noise here. On the right is an HMBC spectrum, which takes 34 minutes to acquire, as it's a less sensitive experiment than the HSQC. You'll notice there's a lot more peaks here because the HMBC shows the correlation from a proton to a carbon several bonds away, and it provides powerful information about the structure and connections within the molecule. For example, here, let's focus on the proton attached to carbon 8, and you can see the couplings to all the neighbouring carbons, including to the quaternary carbons at positions 2, 3, 5, 7 and 17. These carbons did not have protons attached and therefore have no signals in the HSQC spectrum. I mentioned before how critical sensitivity is for NMR spectroscopy, and I want to illustrate this a bit further with this HSQC experiment. I showed before the 250 millimolar brucine HSQC took 8 minutes to acquire. The reason it took 8 minutes is because I needed at least 256 steps in this F1 dimension here to get a sufficient resolution. If we decrease the concentration to 50 millimolar, so it's a factor of 5 less than before, we find a time of 68 minutes is required to be able to identify all the same signals in the spectrum with the same kind of sensitivity. So let's work out what that concentration represents. A 50 millimolar, and using the molecular weight and the sample volume, shows a total mass of 6 milligrams. That's a remarkably small amount of material to acquire such a useful spectrum in just over an hour on a benchtop NMR spectrometer. However, the benefits of high sensitivity do not stop there. Coming back to the 250 millimolar sample, we can make the experiment even faster by reducing the number of steps in the F1 dimension. Instead of 256 steps, we can acquire just 64 steps and reduce the time to 2 minutes. However, you can now see that we don't have enough resolution in this F1 direction, even though we have plenty of sensitivity to detect the signals. And this is a perfect place to introduce the technique of non-uniform sampling, or NUS. The method works by acquiring a particular selection of 64 steps from the 256 step experiment and using a special type of reconstruction algorithm. So here you can see we've acquired an NUS HSQ-CME, and we acquired just 25% of the 256 steps, and therefore this particular implementation takes just 2 minutes. But the results are simply incredible. You can see all the same correlation signals we had in the 8-minute HSQC with the same sensitivity that we had before, but the experiment now only takes 2 minutes. I have to mention that the reason this works so well is because we're relying on the spectrometer having sufficient sensitivity to acquire enough signal in the reduced time available for the NUS experiment. Another technique that can potentially speed up your experiment is the NOAA sequence, which collects an HSQC and an HMBC without having the necessity of a relaxation step in between them. This clever strategy was first published by Kupche and Claridge and can acquire up to 5 experiments embedded in a single relaxation delay, providing a really significant time saving. To test this sequence, we've used a 250 millimolar lidocaine sample, which has just a few signals to follow. Using a NOAA sequence, we acquired both an HSQC and an HMBC. The HSQC from the NOAA is shown on the right, and you can see it looks identical to a traditional HSQC acquired on the left with the same parameters. Comparing now the HMBC, we see no loss in sensitivity of our HMBC compared to a traditional HMBC acquired with the same parameters. But notice, however, we took 21 minutes with the NOAA sequence to acquire both those spectra, whereas each individual spectrum was taking about 17 minutes to acquire. Now we have seen what's possible with 2D NMR methods, I want to come back to the 1D carbon-13 spectra. One of the unique properties of carbon-13 NMR is that it's very similar irrespective of the field strength, while still being very sensitive to changes in the molecular structure. Because of the low abundance of carbon-13 nuclei, it's very rare that a carbon-13 atom has a carbon-13 neighbour, which means the proton-decoupled NMR spectra are nearly always a series of singlets like this. This makes these spectra ideal for identifying and confirming structures. And on the right, I have 2 carbon-13 spectra of cholesterol. The top one here acquired on a 300 megahertz superconducting magnet, and the bottom acquired on a 60 megahertz permanent magnet. And you will see all the peaks are in exactly the same position between the top and the bottom spectra, except for the multiplet of the deuterated solvent. Unfortunately, the low abundance of the carbon-13 isotope also means these spectra are harder to detect, and we really need a good sensitivity to be able to acquire carbon-13 spectra on a reasonable amount of sample material in a reasonable time. So you'll recall our 250 millimolar brucine sample took about 120 minutes, and this was about 30 milligrams of sample. To try out a carbon-13 peakless search, we can use the publicly available C-Search server available on the web at c13nmr.at, which is provided free of charge by Dr. Wolfgang Roben at the University of Vienna. As well as the brucine data, we acquired a carbon-13 spectrum of strychnine, which is structurally very similar to brucine, differing only in the 2 methoxy groups. In this case, we measured the strychnine at 80 megahertz with a 430 millimolar concentration in 51 minutes. The table in the middle shows the carbon-13 peak positions, automatically picked from the spectra. Note that for brucine there are 2 carbons at 42 ppm, which we could not resolve, so we identify 22 peaks in the spectrum, although the molecule has 23 carbons. The peak lists can be entered directly in the web page, or in this case we uploaded the peaks directly from the Spinsolve software via a script. Once the peaks are picked, we get this web page to check the results in about 5 to 10 minutes. You can see the server has over 550 million spectra to search through. Below this we can find the best match, and we see it is indeed brucine. We can see the predicted shift values agree very nicely with our experimental values, and we can see further details of the simulated shift for each carbon site. The results for strychnine were the same, with the best match as strychnine. I want to turn now to the other product I mentioned earlier, the new Spinsolve Multi-X. We discussed how improved sensitivity enables you to measure other X nuclei, such as carbon-13. Well, the new Multi-X model contains a specially developed electronics that is capable of handling multiple X nuclei. On the first channel, we can still acquire proton and fluorine, as we can on all Spinsolve models, while the second channel enables the possibility to measure multiple X nuclei in a fully automatic way. The fact that this is done automatically means there's no need for manual retuning or matching, and no need for recalibration of pulse sequences when switching between nuclei. The automation means the sensitivity of each channel is always optimized, and there's no chance of losing sensitivity due to a manual tune and match that was not done correctly. The automatic switch is instantaneous, so there's no delay, and this means it can work perfectly with our auto sampler, without requiring any operator to be nearby to retune when you want to measure different nuclei. Now, a really excellent demonstration of the Multi-X can be found on this video here, where the Multi-X is combined with our auto sampler and a number of samples are measured and loaded, including this ionic liquid here, which contains proton, fluorine, carbon and phosphorus nuclei. Those 4 measurements were all acquired on that sample in just 1 minute. And you can see the results here. Here's the 1D proton, the 1D fluorine, the 1D phosphorus and the 1D carbon, all acquired automatically without any user intervention and without any loss in sensitivity. This automatic switching technology brings a number of advantages over the old manual tune and match method. It's not just faster and more convenient, but it also eliminates the risk of user error. A manual tune and match might result in lower sensitivity performance if it's not done correctly. With the automatic method, the sensitivity is always optimized, no matter how many times you switch between nuclei. This could be particularly useful if you're using external calibrations for quantitative methods and you had to switch to other nuclei between measurements. And as shown before, it's essential to have this automatic technology if you want to use it with an auto sampler. I've got another example here where we're using a Multi-X for structure verification of an S-Phos ligand commonly used for metal catalysts. The Multi-X system has been used to measure 1D proton and 1D carbon spectra, and we show here it can also measure HSQC and HMBC spectra as well. We can then switch to phosphorus and measure the 1D phosphorus spectrum and also a 2D proton phosphorus HMBC. Phosphorus spectra are particularly useful to identify residual impurities. Due to the excellent sensitivity, we can see here we have an impurity on both the 1D spectrum and on this 2D HMBC spectra. The combination of excellent sensitivity and the Multi-X is also useful for reaction monitoring applications. In this example, we have an organocatalytic reaction using boron and carbon NMR. This is an enantioselective CBS reduction of acetophenone to its corresponding alcohol. A chiral boron containing catalyst is used where the first step in the reaction is to activate the catalyst using a boron THF solution. So now, using boron NMR, we can follow the activation process. In the first spectrum, we see the pre-catalyst. In this spectrum here, we can see the boron THF solution. And in this boron spectrum here, we can see the activated boron species, as well as the boron, the boron THF, and also the monomeric and dimeric boron methyl signals here. Now that the catalyst is activated, we can look at the reaction itself with carbon NMR. So in the first spectrum, we've acquired a spectrum of the acetophenone starting material. And in spectrum B here, we can see the end result of the reaction. The carbonyl has completely disappeared. And you can also see the symmetry breaking due to the boron complex, where the 2,5 signal is now split into a 4,5 and 2,3 signal. Another powerful feature of NMR spectroscopy in general is the ability to access information about molecular dynamics and motion. The pulse gradient spin echo experiment shown here allows us to measure the molecular self-diffusion by attenuating the signal with increasing pulse field gradients. We measure the slope of this line here. It gives us the diffusion coefficient when we plot it on a Sesquill-Tanner plot. Now, having good sensitivity is important because we're attenuating the signal, and it also enables us to combine this technique with other nuclei. So in this example, we're measuring a lithium-based ionic liquid that's often used in experiments to measure the transport properties of battery components. This sample has one anion containing fluorine and two different cations. One is lithium and the other contains hydrogen. This means we can carry out that PGSTE diffusion experiment on a Spinsolve configured to measure these three different nuclei, and we can separately determine the self-diffusion coefficient of those three different ionic species in the mixture. And we can then, for example, study how those self-diffusion coefficients would change as we change the concentration of the components, or even change one of the components themselves. In this final example here, I want to show you some measurements made using silicon NMR. For chemists studying polysiloxanes, the use of silicon NMR spectroscopy is extremely useful. However, it can be challenging as the sensitivity of the 29 silicon isotope is similar to that of carbon-13. However, with good sensitivity, we can acquire very nice spectra of different polysiloxane samples. In the sample on the left, we have a polydimethylsiloxane co-methyl hydrosiloxane with trimethylsilane terminations. The 29 silicon depth spectrum is shown on the left here, and the peak positions of the different building blocks in the polymer correspond very nicely to the positions given in the literature. If you are unsure about the peak assignment, you can use a 2D HMBC, such as the one shown here on the right. This is measuring a polydimethylsiloxane co-methylphenylsiloxane with trimethylsilane terminations. You can see very nicely which protons are coupled to which silicons, and in addition you can see the small size of the aromatic signal shows that this copolymer is largely consisting of the dimethyl blocks. In summary, we've seen that the ongoing and increasing performance of the Benchtop NMR spectrometers is opening and enabling more and more applications. The convenience of a cryogen-free magnet that can sit on the bench can be realized only if the spectrometer has the sensitivity, resolution, and capabilities necessary for the sample the customer is measuring. We have shown the increased performance available with the new Spinsolve 90. That is a direct result of maintaining the magnet homogeneity as we increase the field strength. We have also seen the range of applications the new Multi-X enables, and in particular how sensitivity enables us to measure more complicated molecules faster than ever before on a benchtop NMR spectrometer. Well, thank you very much for your attention, and I look forward to answering any questions you have. Thank you.
Thank you, Andrew, for that great presentation. We now have time for some questions. One question that came in, someone asks, can phosphorus 31P NMR be acquired with proton decoupling?
Thank you, Anne. Yes, the answer to that is yes. We have sequences for phosphorus acquisition with proton decoupling. But actually, all of the spectrometers can measure proton and fluorine. So we also have sequences for doing phosphorus acquisition with both proton and fluorine decoupling. And even we can also do a proton-acquired spectrum with phosphorus decoupling. So sometimes you get some large coupling in your proton spectrum. So all of those sequences are all set up on the spectrometer. You can just go into the menu, hit a button, and it's all set up and configured, optimized to run.
Great. Thanks for explaining that. Someone else asks, is it possible to run NOD or non-deuterated NMR?
Non-deuterated NMR, that's a really good question. So the answer is yes. We don't need to have deuterium solvent in order to lock. So the locking in the system is done with an external lock that's completely separate. So it means you can acquire spectrum in solvents which are deuterated or non-deuterated. Now, of course, if you use a deuterated solvent, you don't have any solvent peak in your proton spectrum. If you use a non-deuterated solvent, could be water or normal chloroform, for instance, you will see a large proton peak, a large peak from that solvent, and that's where the solvent suppression examples I showed earlier on are so useful, because it means you can run NOD NMR. If your peaks are close to the solvent peak, then you use solvent suppression to suppress that strong solvent peak. But you can certainly run without deuterated solvents, yes.
A related question talking about solvent suppression. Someone asks whether the Spinsolve ultra spectrometers can perform solvent suppression with solvents other than water.
That's also a great question as well. The answer to that is yes. You can run the solvent suppression sequences with many other solvents. One of the really important things to keep in mind is that other solvents may have more than one peak in the proton spectrum. So water and say chloroform are quite nice. They just have a single peak that you need to suppress. The solvent suppression sequence we have on the Spinsolve can actually suppress up to 3 different solvent peaks. You can even configure it so that it will automatically find those peaks to suppress. And one further thing which I didn't mention in the webinar is it's often useful also to run those sequences with carbon decoupling. So if you have an organic solvent, let's say ethanol, you might have 3 different peaks that you want to suppress, but you also want to suppress the carbon satellites because they may be as large as some of the analytes that you're trying to measure. So yes, you can run with other solvents, and up to 3 peaks can be suppressed with the sequence that we have built in there in the Spinsolve.
Thank you for that answer. Someone else mentioned the fact that shimming is common for high field NMRs. Is shimming required for the benchtop NMRs? And if so, is it manual or automatic or both?
That's also a really good question as well. So the short answer is yes, we do shimming, and that's done automatically. We have some automatic sequences set up to do that. One of the sort of big differences here between a benchtop NMR and high field NMR is while on high-field NMR you need to shim essentially for every sample you put into the spectrometer, especially if there's a change in the solvent. With benchtop NMR, you often only need to shim once, and you can then measure a whole series of samples, one after the other, without needing to re-shim in between them. The automatic shim, in any case, typically takes 3 or 4 minutes. And you can run that shimming sequence on either a reference sample that you can put in, and in fact you can also run it on normal samples as well. So you can, if you put a sample in and you think, oh, this maybe it does need to be shimmed for this sample, you can actually shim on the sample itself. This is particularly useful if you're doing reaction monitoring, where you've got a reaction running for a long time and you're measuring one spectrum after another. You don't have the chance to put a reference sample in there. You can actually shim on the reaction mixture that you're measuring. So yes, we do shimming on benchtop NMR, but not nearly as often as you need to do it at high field NMR.
Great, thank you for that answer. And sort of a related question, you mentioned the shimming routines and things. Someone asks whether to improve homogeneity you would spin the sample rather than shim.
Right. Yeah. So that's again what everybody is used to from high field NMR is you insert the sample into the spectrometer, and one of the first things you have to do is wait for sort of 30 seconds while the air starts the sample spinning, and the reason you spin the sample there is to sort of average out some residual fluctuations in the field. Now with benchtop NMR we don't do any spinning. And there's a couple of really important reasons for this. The first thing is that the spinners are often, one of the more fragile elements of a system. So if we want to build a spectrometer that's being used in a lab in a very sort of routine, robust way, if we had to spin the sample, it would be like a weak point. It would be something that would be prone to failure, which wouldn't be very useful. It also means you would need to have a source of compressed air as well running to the instrument, so it would sort of add more complications to how you site and run it. So that's why, as I sort of spent some time talking about in the webinar, we really put a lot of emphasis on shimming the magnet itself. So the magnetic field that we generate from our magnets is very uniform. It's very homogeneous anyway. And because we can achieve that homogeneity, it means when you put the sample in, there's no need to spin, and the system will work in a very robust way because of that.
Great, thank you for that explanation. Someone asks, is it possible to use quantifying the molecule or QNMR?
Yes, so QNMR, quantitative NMR, is something you can absolutely do on the Spinsolve benchtop NMR. So one of the intrinsic sort of features of nuclear magnetic resonance spectroscopy is it is an inherently quantitative method. So, and in fact, if you're measuring, for instance, a methyl group, it doesn't really matter what molecule that methyl group is attached to. You'll get the same signal from that chemical group if you integrate it. So it means you don't need necessarily to have reference standards in order to do quantitative NMR. You can compare one peak with another peak in the spectrum, or you could have an internal reference which could be a different molecule. And you can also do QNMR with external reference samples as well. So you can measure one reference sample, and then you can measure your samples of interest as well. So there's a number of different approaches to QNMR. It works very well on the Spinsolve benchtop NMR. We've really been so impressed with the quality of the data that you can get, quantitative data you can get. There is actually, just mentioned in the software, a dedicated QNMR module where you can set up a method, you can sort of predefine all of the acquisition and processing steps that you want to do, so that you can then just repeat the same method, exactly the same. It has it has an audit trail in it for people working in regulated environments and digital signatures. So QNMR is absolutely something you can do, and very, very useful, and it works well.
Great, thank you. I know you mentioned an example of a polymer application. Someone asked whether you can elaborate a little more on whether the Spinsolve 90 can be used for polymers.
Yes, absolutely. So, I mean, probably the best place I can direct you to in terms of polymer applications is on our website. There is a whole section on polymers under the applications, and then there's a number of very interesting polymer blog posts where one of our application scientists has gone through a number of different sort of applications that you can use Benchtop NMR for. So, I mean, just to name a few, one of the applications is to get at the molecular weight of the polymer. So you can sort of identify the end groups, which obviously you always have two end groups on a polymer, and then you can compare that signal to the signal of the whole backbone of the molecule, and then from that ratio you can work out how many units are in the polymer, or what the molecular weight of the polymer is. There's also another very nice example doing copolymer composition, so using a Spinsolve to do copolymer composition. And in fact, also you can combine the pulse field gradient experiments that measure diffusion with polymers. So that can be very interesting, because polymers, as large molecules, they have different self-diffusion coefficients depending on how large they are and what kind of polymer they are, if they're branched or not, and their molecular weight. And so again, there's a nice blog post there showing the combination of diffusion NMR with the pulse field gradients and polymer samples to look at different samples and characterize the difference between those polymers.
Great, thank you very much. Another question that's come in is it possible to do neat carbon-13 experiments?
Neat carbon, so if you had, I think the question means if you had a neat sample, for instance, I mean, you could have a solvent or something, and you can measure it. The only requirement is it needs to be a liquid, and you can absolutely do carbon-13 NMR. So, in fact, I think we have a series of very nice experiments where you use a series of different alcohols. It's an education experiment that we generated for our customers who work in chemistry education, and you can actually measure. So an alcohol carbon-13 you can actually measure very quickly. I think in 4 scans or 8 scans you can see a very nice spectrum there straight away. So they're very quick experiments to do if it's a small molecule. Even doing carbon-13 NMR, I mean, that shows you how useful good sensitivity is. And carbon-13 is actually, NMR is a good introduction to NMR. They're a little bit simpler to understand sometimes than a proton NMR spectrum for someone who's new to NMR. You can directly count the number of carbons. They're all nice singlets. They all appear in very particular places in the spectrum, depending on what kind of molecule it is. So yes, absolutely. As long as it's a liquid, you can measure carbon-13 NMR.
Great. Well, thank you, Andrew. Someone else asks, is there any special maintenance required when working with a Magritek Benchtop NMR?
That's a really good question. So obviously one of the things we've worked very hard to do is to make sure that this is an instrument that can work in a normal chemistry lab, and it does not have all of the sort of special overhead that many of us are used to when it comes to high field NMR. So there's obviously no cryogens and no compressed air and none of the other things that you would normally associate with high field NMR. But actually the instruments themselves have no moving parts other than a couple of fans on the on the electronics. And you have to remember, with NMR, the sample never touches. We never touch the sample itself. It's always in the NMR tube. So it's not like we need to clean the inside of the instrument or anything like that. So in fact, the benchtop NMRs will run for years and years and years without any real maintenance requirements. I mean, you can give it a clean and get the dust off, and sometimes you need to clean the bore itself just because a bit of dust got down there or some grease or something. But other than that, there is just very little. They're very robust instruments, and they keep running and running.
Great, thank you. Someone else asks about some of the features that you've mentioned and wants to know if these are all available on a single instrument, things like ultra suppression, 90 megahertz gradients, multi-X, etc.
That's an interesting question, actually, and there are we have many of the features available on single instruments. Obviously, we have different magnets, so we have 60, 80 megahertz, and now the 90 megahertz magnets. On the 60s and the 80s, we have the ultra, we have the multi-X, and we have the gradients, and they can all be combined. So you can have, for instance, an ultra multi-X that's measuring a range of different X nuclei. So that's all possible. With the 90 megahertz, it's our newest product, so we're working on those features, and we intend to have those features available in due course. So absolutely, the intention is there that you can bring them together.
Great. We have another question about some of the capabilities. Is the NUS processing part of the instrument software? If so, which algorithm is used?
Yes, so that's in the latest version of the software. We have the NUS acquisition as well as the NUS processing. I don't know exactly the algorithm that's being used. I think it's a variation of the iterative soft thresholding, but I'd need to. I'll have to get back in touch with the person who asked that question later. But I think that's the algorithm that's being used. But it's again, the idea is we really want to remove a lot of the complications of having to run NMR spectra, so we don't expect the person operating the instrument to be an expert. They really should just be able to go into the protocol, push the button, and they can get advantage of these NUS methods, which are really quite exceptional. And both the acquisition and the data processing is all handled within the software. That's not to say if you have your favorite algorithm, we also have the raw data available, so that is, the raw data is available for you to process with your own software as well, if you'd prefer to do that.
Another question that has come in is someone asks whether an auto sampler add-on is available for either the Spinsolve 80 or 90.
Oh yes, absolutely. So we have the auto samplers available for all of our models. So for the 60s, for the 80s, and for the 90s, we have a 20-slot auto sampler. It's very robust and very affordable, and we've been exceptionally pleased with how well it works. It, you can set up a whole batch of experiments. There's a very nice queue interface where you can set up a queue with multiple samples, multiple experiments for each sample, and you can set that queue running, and it will switch between the different samples. And as I mentioned in the part about the multi-X, if you have the multi-X feature, it will also switch automatically between the different X nuclei as well, without any, no need to intervene at all. So yes, there is an auto sampler. It works with all the models, and we think it's a very, very nice add-on for people who want to run batches of experiments, especially running experiments overnight, for example.
Someone else asks, can the multi-X detect more than one nuclei shown? For example, aluminum-27.
Yes, so that's a great question. So, I mean, in principle, it's from a hardware point of view, it's just a matter of where we're setting up the system to operate on. So there's no sort of intrinsic limit in the hardware there. But what I would suggest to that person is we'll get in touch with you and look at the particular nuclei that you want to measure, as long as there's an NMR active nucleus that we can measure. And also remember what I was saying about sensitivity, as long as there's sufficient sensitivity to measure the samples they're interested in, and that's something we can help anybody, look at that question and understand. But as long as that's feasible and it's going to work, then we'd be very happy to talk that through with them. I mean, the offer is there for customers to talk to them about their samples, do test measurements, and always we're very conscious for many people. The question is, will benchtop NMR work for my particular application? And there's nothing, the only way to really answer that question is to go and measure some samples and actually see what happens, because every molecule is different.
Great. Thank you so much, Andrew. I'm afraid we've run out of time. We do have more questions outstanding, but those will be passed along to the speaker and for follow-up. So thank you again, Andrew, for your great presentation. Thank you to all the participants for being a great audience. Please be sure to check C&EN or C&EN Online for information on the next edition of C&EN webinars and for the replay of this webinar. Our thanks to On24 for technology and production services, and thank you very much to Magritek for the sponsorship that made this interactive webcast possible. For C&EN webinars, I'm Ann Thayer. Hope you have a great day.